Speaking of cell cycles, we do have checkpoints that control the transition between phases of cell cycle. This process is regulated by cyclines. That's one. Second is cyclin-dependent kinases, and then third part is the tumor suppressors. So basically, the cell cycle do have multiple phases like M phase, G2, and there's G1, G0, and S phase. So all of these are regulated by the cyclines and the cyclin dependent kinases with the tumor suppressors. So for the M phase, it's the shortest phase of the cell cycle and includes two things. It includes the mitosis and also the where is it? Cytokinases. Okay, so first is the mitosis, and the second is cytokinases. These are two parts that they're included in the M phase, and it's considered um, the shortest phase of the cell cycle. So for the mitosis, it's made up of multiple steps. That's the prophase, prometaphase, metaphase, anaphase, and telophase. So all of these are within the mitosis. And for the cytokinesis, it basically means when the cytoplasm of the cell splits into two. So basically, cell cycle is just like the progress of a cell. So it may end up just being two cells instead of one. We also have the G1. It's of a variable duration. So where is G1? So this is G1, and it could be of a variable duration. We do have a note over here, and it says that the transcription occurs throughout which cell cycle. It happens in the interface that is between GS and G2. So basically, we do have the transcription, that is the change that occurs from a DNA sequence into a messenger RNA, and that occurs in the interface. Okay, so far so good. For the regulation of the cell cycle, we do have, as we said, three main regulators. The first one is cyclin-dependent kinases. They're constitutively expressed but inactive when not bound to cyclin. So they're always there. They're like always existing, but they're considered inactive. They do not work if they are not bound to cyclin. So there is a need for them to be bound to cyclins in order to be able to function. That's for the cyclin dependent kinases. And that's what this name applies because they depend on cyclin in order to work. Also, we do have the cyclin CDK complexes. It says that cyclins are phase-specific regulatory proteins. Phase-specific regulatory proteins. Okay, so they do regulate, they are proteins that they do regulate stuff depending on the phase, like they choose a specific phase to work on. They do activate CDKs, and CDKs should be, okay, research web, then, okay. Cyclin dependent kinases. Okay, good. That's just the cyclin dependent kinases. Um, when stimulated, they do activate the cyclin dependent kinases when it's stimulated by growth factors. So they do um, that activation process only if they were stimulated by the growth factor. So they don't do it on their own, just like this. They need the growth factor in order to be able to activate the cyclin dependent kinases. Okay, so the um, cyclin dependent complex can then phosphorylate other proteins. So the main idea is that these are a combination of two main um, things. The first one is the cyclins, just like something called cyclins, as we said over here. It was a component of the cell phase. And then we do have the cyclin dependent kinases. So these two parts joined together, they form complexes. And these complexes are like identified as proteins that they do regulate specific phases. And they also activate like one of their parts, that is the cyclin dependent kinases, but that happens only if they're stimulated by growth factors. So it says that these complexes then can also can then, after they are stimulated by the growth factor, they do phosphorylate other proteins, just like the RB protein, the retinoblastoma protein, to coordinate cell cycle progression, that to help with the cell cycle progression. This complex must be activated or inactivated at appropriate times for cell cycle to progress. So we need to be like the body needs to be really specific and accurate when it comes to time in order to decide to activate those or inactivate those in order for the cell cycle to progress normally without any problem. We also have the tumor suppressors. The tumor suppressors, just like P53, that leads to P21 induction, and that leads to CDK inhibition. And we said that the CDK is just cyclin dependent kinases. So P53 causes an induction of P21, and then the P21 causes like CDK inhibition, and that would lead to RB hypophosphorylation. So if it causes hypophosphorylation, that means it's activation for this specific RB. Of course, that has hyperphosphorylation. That activation leads to G1S progression inhibition. So it causes an inhibition of the cell cycle. So it does not move from the G1 to the S phase. And if we take a look over here, so this is the G1, and it should, like the cell, this is the cell, should move like from the N phase to the G1 and from the G1 to the S phase. But what happens if the P53 modulates this area over here and it does not allow to the cell to move to the S phase? Okay, so it's considered a G restriction point. Both of these actually. So it's P53 that is activating the RB, but they're both considered modulators of this point, causing a restriction over here. So, of course, a mutation in the tumor suppressor gene, a mutation over here of the regulator of this point, it results in an unrestrained cell division. So the cell keeps dividing 1 and 2 and 3 and 4 until we do have some kind of tumor or whatever, but it's not normal. So, for example, is the Lee-Fraumeni syndrome. Okay, so we also have the growth factors, just like insulin, PDGF, EPO, and EGF. They bind tyrosine. All of these growth factors, they do bind tyrosine kinase receptors. So to transition the cell from G1 to S phase. So there we do have some receptors that are they're called the tyrosine kinase receptors, and they uh, are activated by the growth factors within the body, just like these growth factors. They these receptors, they do regulate the transition of this cell, like the dividing cell, from the G1 to the S phase as well. So over here, also from the G1 to the S phase, and that's where the growth should go. Okay. And if we take a look over here to see exactly what we have. So we have the M phase. That's where the mitosis occurs, and also the cytokinesis. For the mitosis, we do have a note. It says that cell division, DNA condenses to form chromosomes. So within the mitosis, the DNA condenses, of course, to form chromosomes. And then vinca alkaloids, just like vinblastine, vincristine, vindicine, and vinorelbine, and also taxanes, just like uh, paclitaxel and docetaxel. All of these are existing over here. That's the vinca alkaloids. plus the taxanes. They do exist over here within the mitosis. So the DNA condenses to form chromosomes, and it does so because it's going to um, divide. Yeah, so that's for the M phase. And then we also have the G0 phase over here, where the cell goes out a little bit. And then we have the G1 phase. Let's see if you do have a note. For the G1, it's the pre-synthetic growth. So what happens in the G1 phase, it's the organelle duplication. So the organelles within the cell itself is going to divide and multiply before the division of the cell. So that's for the G1, okay? And then we said that we're going to see these proteins deciding whether the cell is going to move forward with this division or not. And then we go ahead to the DNA synthesis. Um, that is the S phase over here. So the S phase is actually the synthesis, that's the DNA duplication. So what happens is like the actual DNA and the histones, like the histone, the circular uh, kind of ball thing with the DNA wrapped around, is going to duplicate and also is going to form anti-metabolites. Just not going to form the anti-metabolites are just some kind of a class of drugs that they do work by interfering with the cell division, like the DNA constituents of the cell. So they will inhibit like the cell, the DNA division and stuff like these. So that happens all within the S1. So some examples of these antimetabolites are the fluorouracil, uh, mercaptopurines, and methotrexate. Yeah. And also we have the G2, so that's the last part of the cell. And it has some kind of notes. It says is the pre-mitotic growth. So that's before the division, like the actual division of the cell from 1 into 2. So that's the G2. It could have some DNA damage repair over here. So if you do have a problem with the DNA, it could be repaired over here within the G2 phase, just like by using the nucleotide excision repair. Some drugs also can work over here. They're like bleomycin and doxorubicin, which is a chemotherapy. So if we take a deeper look, then we start over here by like this genetic material, like from one cell, that's the G0. It goes through the G1 to the S phase, and it still has the same genetic material. And then it starts to form two distinct stuff. And these two distinct stuff are going to make it to the M phase, which means like the last division of the same cell into two different cells, and each one of those goes uh, you know, into her own cycle. Plus, what do we have over here? The rest of it is just a presentation of all the things that we have just talked about. So basically, it says that let's assume that we do have a DNA damage to the cell, and of course, it does have a DNA damage. It should not proceed with the replication process. So the DNA damage is going to notify the P53, that's the regulatory protein. So if it is activated because it senses the DNA damage, then it would activate like this green plus sign means activating the B21. That B21, if it's activated, then it would inhibit the whole process of dividing the cell and going through extra steps that the cell need to do in order to decide dividing or not. So if, like we said, P53 would activate the P22, and that would inhibit this whole process. And one of those things is the RB protein, and we do see that it does have like phosphate groups. So just as we said over here, that it would be less activated if it is phosphorylated. Is that what we said? Actually, it was the cyclin dependent complex that's the thing that can phosphorylate the RB protein, and that's true as a matter of here because that complex is the one that is going to phosphorylate the RB protein. So that's for now. Just in case this patient does have Li-Fraumeni syndrome, and that's the loss of function, or also if someone is infected with human papillomavirus, which has E6 protein, and this E6 protein interrupts the P53, just like the Li-Fraumeni syndrome, it would interrupt the P53. So it would disrupt that function, and it just keeps the cell dividing and again and again and again. Of course, one of the other things that the P53 can do is just inhibiting this BCL2 and BCLXL, leading to the whole process of caspase activation, and that means the apoptosis. That's the intrinsic pathway of cell death depending on itself. So the caspases within the cell are going to be activated and then kill the cell. And that should be it for page 44.